And so David is working on a Gates Foundation project there and looking at on-farm sparse testing, which I'm sure he'll be able to talk to you about in a minute. So welcome, David. Thank you for coming. Are you happy to take questions as you're going through the presentation?
Yeah, I think yes, because I'm going to talk plumbing stuff and maybe.
Yeah, so please stop him. Yeah, lovely. And so the way it'll work is David will go first for about 30 minutes and then Dom will have the second half. So we'll take questions throughout and then also during the break too. So,
yeah, thank you. Thank you, Daniel, for the great introduction and for organizing this. Also, Hans. And this is a pleasure to me to stay here and share this a little bit of knowledge we are getting from the CG. So I did this work. Also, Chris was involved in this this work that I will present, and of course, again related to plant breeding, specifically for hybrid breeding crops, for a specific case of maize hybrid breeding. And we are proposing this new strategy that offers breeder a balance between resource efficiency, higher accuracy, and simplicity to implement this. So let's move. So maize hybrid breeding, well, breeders do cross parents that are from different pools in this case to exploit the heterosis to create hybrid. This is basically. So we have normally, I would try to explain this hybrid breeding scheme to help you a little bit of background. Normally they have two pools, so heterotic pools, and within each pool they create inbred lines here and then start the evaluation for selection and also for deliver new hybrid varieties. So basically they use recurrent reciprocal selection methods, but they do this kind of multi-stage selection process. So they start with first stage, stage 1, and they test all the candidate lines with one tester. They do preselection and they move to the next stage successively or subsequently, and they increase the number of testers as the stage are moving. And they do the test cross evaluation at every stage, but of course it will be reduced like a bottleneck and ended with very few selected material. Yeah.
For people who will rewatch this,
yeah,
it's really helpful if you use the mouse pointer to point out things.
Oh, perfect.
Because they
Yeah.
don't see you on it.
Perfect. Thank you. So they do this reciprocal recurrent selection, and the selection is based on the general combining ability by testing the candidates with tester. We call the tester, and the tester is coming from the opposite heterotic pool or the complementary pool. So this is basically the population improvement component in a hybrid breeding scheme. And then in later stage, they still continue and advance with very few candidates, hybrids candidates, what we call the product level component that is focused on the specific combining ability. So they try to identify the best performing hybrids, but also identify hybrids with marketable traits like stability, for example, or high yielding, but also disease resistant. They can also make some amendments, including or introducing some resistant genes for hybrids to be stable across many environments. And they do this testing, well, in a broad number of or high number of environments in very late stage, but in the early stage, they do in very few environments. And the reason why they are using this multi-stage process, there is some reason that I will explain next. You might also wonder that this approach or this multi-stage selection process is extending this breeding cycle because it took several stages until they decide or they feel confident to recycle the parents for to make the new crosses. So why not recycling the parents early, like in the first stage or early stage? The reason is because there are some challenges. Basically, early stage is characterized by a large number of candidates. So the ideal design for evaluating or estimate the general combining ability will be a complete factorial design that involve testing all the possible hybrid combination from the two pools. But the problem is that if we have a very large number of candidates on each pool, imagine we have 1,000 candidates on each group, then we will have millions of hybrids that will be impossible in practical to evaluate. So what the breeders opted to do instead, or to address this situation, is, well, do this multi-stage process. We start evaluating all the candidates only with one tester. So they do kind of pre-selection, and they reduce the number of candidates as they advance in the stage, and they increase later with more tester. But of course, the highest selection intensity happen at the early stage with only one tester, and then of course they increase numerous tester in later stage, but only for few candidates after selection. So the problem with one tester for doing this preliminary assessment is, well, it poorly represent the heterotic pool. So few haplotypes from the complementary pool are being exploited with one tester, which is not enough to accurately estimate the general combining ability. And also it puts a lot of pressure in the first stage only with one tester, which is suboptimal for selection and advance to the next stage. Well, with the advent of genome-wide markers and genomic selection, we have some opportunities, and people have been thinking how to address this situation, and they proposed to use an incomplete factorial design. So instead of doing all the possible hybrid combinations, they just took a sample of the incomplete factorial design, for example, and they showed that they can increase the prediction accuracy, also increase the genetic gain with these designs. However, it still has some practical limitation. It requires hand pollination that is very labor intensive, also costly and challenging, especially with we need to synchronize the flowering time between the crosses, because if we need to make a specific crosses, and if there is a lot of variability of the flowering time between them, it will be very challenging to make that crosses. So I cannot say this is impossible, but for some breeding programs, small breeding programs or public breeding programs, it will be still a challenge to implement this kind of design. So in DCG, our breeders in Mexico, they tried to implement this incomplete factorial, but was very, very challenged, and they failed to make many of the crosses, so they couldn't make all the crosses that were proposed. So we were thinking how to address this, and we proposed this Sparse test-crossing strategy for the early stages, using few testers in sparse designs, of course leveraging the genomic relationship matrix to connect the candidates' genotypes. And this idea comes from the Sparse testing idea that we are more commonly used in DCG, this Sparse testing strategy for phenotyping. And the idea is basically that traditionally the breeders, they want to evaluate all the candidates in all the environments. And always they have to balance, so there is a trade-off between the resource and the accuracy they want to get. But using this sparse testing, we have subset of candidates evaluated in different environments, so they can make a little bit more efficient and evaluate more candidates with this sparse testing strategy. And we try to translate this in the concept of the tester. You can see like the environments like a testers. So the environments become like testers in this Sparse testing design. So this Sparse test-crossing design, basically, well, we create subset of candidates. It might be just at random, And each subset of candidate can be crossed with different testers. For example, it might be 3, it might be 4 or 5. We will see later how we can decide the number of testers, but use, for example, 3, then you create a random 3 subsets, and each subset will be crossed with 3 different testers from the opposite or the complementary pool. But importantly is that these full SIPs get also sparsely distributed among the testers, like in this diagram. And the tester also can be just randomly sampled from the complementary pool. Each candidate is crossed with only one tester. So in this is to keep the testing resource fixed compared to the conventional design with one tester. So we keep the resource fixed, but we evaluate or use few testers to estimate the general combining ability. And of course, now we use the URM to connect all the candidates and exploit this the complementarity, the haplotype complementarity between the groups. What is the hypothesis behind this? Well, having more testers, we have a broad sampling of the haplotypes from the complementary pool. Because the idea is try to sparsely distribute the tester among these full SIPs. Full SIPs have common haplotypes, and these common haplotypes are tested with different haplotypes coming from different testers. So we have a better evaluation of the complementarity. And well, this is the advantage of genomic selection. Then the genotypic predicted effect of one genotype will reflect the average performance of the multiple test scores, not just one, just because we have this connected through the genomic relationship matrix. And we expect enhanced genomic general combining ability prediction accuracy with this approach. We did this, well, a simulation study. It was a simulation study. We ran this hybrid breeding scheme. The numbers, well, you can see the number of parents, number of crosses, number of candidates. These numbers are coming mostly from the numbers that the breeders, hybrid breeders in DCG are using, around a bit more than 1,000 candidates. And then we apply and we compare all the Sparse crossing strategies with a conventional one. And we were interested in check what is the prediction accuracy, but also the genetic gain with these Sparse crossing strategies. These are the different test crossing strategies. The first one is the conventional one tester at the very early stage. Use one tester crossing with all the candidates, but then we increase the number of testers from 2, 3, 4, 5, and then increase to 10, and so on, until we include an hypothetical case of 1,200 tested. That means the candidates from the opposite pool becomes like the tester completely. And that scenario, that estimate scenario reflects the structure of the incomplete factorial designs that I mentioned it before, that we are also interested into check. So, well, you can see here, if we have 2 testers, then we split the candidates in 2, and so on for each of the strategies. Okay, let's see some results. Before, I would like to show this. This is the genetic divergence between the heterotic groups, just to show, nicely show how they become split. Of course, this is PCA plots at the different cycles, cycle 1, 2, 3, 5, 10, and 15. Maybe you cannot see, the numbers of the PC1, but as we advance in the cycle, over cycle, the PC increase the proportion of variants explained. So in at cycle 10, for example, we have 70% explained used by the first PC component And we started from a very, very low genetic diversity two pools. Actually, we created one population and we just sampled the two pools from that. And we did use 3 cycles of random crossing and selection just to generate family structure. Yeah.
That's interesting. I think, is that really what you think happens during the breeding program? That these heterotic pools become more?
Yeah, it should happen in a well organized and structured breeding program. Of course, here is simulation is simplified, it's just one trait, and we see that this quickly split the two heterotic groups. In real breeding programs, they work with several traits. So managing all the traits simultaneously, well, it was slow this separation, but in principle, they will see something like this.
But this is genetics. That's a marker PCA, or is that a PCA?
Markers PCA, yeah. Markers PCA, yeah.
Just to add, Mark Cooper and colleagues have published a paper where they essentially show that's productivity over the pioneer germplasm in maize. You know, the two poles are kind of diverging.
Also, we depending how they manage the diversity and yeah, diversity when they select the parents. So, well, but it is just nice to show how that's happened because we also were interested in to see how these strategies perform across these different stages of divergence of the breeding program. That was also interesting to show this. So at the very early stage, cycle 1, when the divergence between the pools is low, you can see 6% explained by the PC1. So we have the highest increase in accuracy by increasing the number of testers. You can see in the x-axis the number of testers and the y-axis the accuracy. So we increase the number of testers, we see some increase in the accuracy of general combining ability. And it was, if we look at the 10 tester, we got around 11%, 10% of increase in accuracy. But we see also that with 3 or 5 testers, we almost reach the same accuracy. And what happened beyond that, even if we increase highly the number of testers, like the incomplete factorial, we still have the same plateau. We don't get any more benefit using much more testers. You see, with 5 testers like here, 10 testers here, and then using all the candidates from the complementary pool as, let's say, tester, then we don't see really too much increase in the accuracy.
Could it be that the diversity within each pool is quite similar? There's all diversity within the pools.
Yeah, the diversity is playing a role.
Because you're testing the same.
Yeah, but this is the trend. At some point, if we get low diversity, we will start to see a plateau, or even we don't get any benefit, as I will show next.
We also try different within population diversities and try to make it a bit more extreme. And we saw that we started with a completely unrelated population with a lot of diversity, and this is just the very first cycle. So just as a consequence of breeding and the numbers we do work with in plant breeding, we pretty quickly get to a level of diversity that is quite well representative of what we see here.
David, why?
Yeah.
So we see increase in accuracy of GCA when you increase the number of testers with genomic models, but not with the phenotype model.
Yeah, well, I wanna talk that just in a few slides, so we will address the reason why. Then, in cycle 10, with the divergence between the pool is high, they are highly separated, we don't see too much, basically, the advantage disappear, and we don't see too much increase by using more tester. And also related to to your comment, there is not the diversity low at that point. And this is why with one tester, we can represent all the variability in the pool. So it's related also to the genetic diversity within the pool that we observe.
I mean, that point, right, is really what happens is that blue point at the bottom of the right is a spread of points in the dimension that we don't see. So there is still some diversity, probably quite some diversity in that point. It's interesting that one tester is not counted.
Of course, this is a simulation extreme case, but this is like the trend in general that we will observe.
So just to add quickly, we're talking about 10 cycles, and in CG breeding, this would mean 30 to 40 years at least.
Exactly.
Obviously, years cycle time still is pretty long, and for breeding program without any introgression, just selecting for one trait that is, well, it's a simulation. It's probably more intense than in reality, but it's not too unrealistic.
So your question, Gregor, what happened with the phenotypic? Well, the phenotypic, as we cross, just each candidate is crossed only with one tester, then we don't get the benefit from the relationship between the individuals. It's just crossed with one tester. Actually, it might generate some confusion because all the lines will be crossed with difference. Then the comparison is not fair to estimate general combining ability. But basically, we are not. Also, you see the accuracy, of course, is lower because we are not exploiting this the URM. We are not exploiting. So there is not.
That's pure phenotypic. It's not a database model.
Sorry, this is just phenotypic, no pedigree. Yeah. So no benefit implementing sparse. Conversely, I think it will generate some confusion because it's only one tester. Well, only we cannot separate. We are evaluating a specific combining ability with the phenotypic selection because each candidate is crossing with different tester.
In that scenario, we have obviously genomic relationship available.
Yeah. Yes.
Is that realistic?
Yeah, it's realistic. It's realistic. Yeah. Our breeders in the CG, they are genotyping now the candidates at the early stage. Yeah. Well, this graph just show how the accuracy decrease in general across the cycle of selection. Well, you can see this one, the red one is the conventional approach, only one tester, and the other ones are using more than one tester, 2 or 3, but you can see the difference between 2 or more testers becomes really small. Within that 3-5 tester, to be sure you can get exploit this disadvantage with the Sparse crossing. Then we run breeding programs, but using selection based on each of these Sparse crossing approach. Also, I didn't mention, but we simulated 3 degrees of, we can say, heterosis that based on different degrees of dominance when we simulated, but we can say heterosis, and we get, of course, more gain if there is more heterosis in the population to exploit. Of course, less when there is low dominance, but we can see 6% for what we can say we have observed more or less in our breeding program for this medium dominance degree, medium heterosis. Okay, so basically we show that Sparse crossing with few testers enhances the accuracy of the estimate, the general combining ability, and that translates in higher rate of genetic gain. With 3 to 5 testers, we think a practical balance between simplicity, resource efficiency, and improved accuracy to estimate the GCA. And that's might enable the rapid cycling for the breeders to cycle parents to the early stage, the first step of agronomic testing, and that potentially short the breeding cycles and boost the genetic gain.
Do you translate in reality how shorter would be the cycle?
Well, I've shown you at the beginning, they do like 3 stages before they recycle the parents because breeders are not confident to recycle at the very last stage because the reason I mentioned. So they do at stage 3 when they use like 4 or 5 testers. They become confident and they select the parents. With this, well, they need 3 cycles to do that, as they are doing, as they traditionally do that, 3 cycles for recycling.
So the question is, how long is the cycle?
How long is the cycle? They can do per year, maybe 2. I'm not really sure about this, but maybe.
You probably could get from around 3 years to 1.5, something like this. So you can talk about a 50% increase.
Yeah. Okay, so we compare with the factorial design and we see that we get better balance between accuracy and resource efficient. If you tester, we get same accuracy that an incomplete factorial design. I didn't show this, but it also, Sparse also preserves genetic diversity by using more tester. We exploit more broad complementarity and helps to enhance the long-term genetic gain. And as I showed in the first graph of the results, just simply using in selected parts based on the general combining ability makes the job to create and separate the heterotic groups. Well, if you want to look some details, you can check also the preprint version and acknowledge some of the colleagues that also contribute to this work. Thank you. I don't know if you can. Yeah.
David, I missed when you're picking your testers.
Yeah.
Established individuals from previous cycles
We did for the current generations.
for the current.
Yeah, for the current generations. Well, there is a situation breeders, they like a lot use very widely used testers. That is a big problem because it's generated some bottleneck using the same tester for several cycles. It has some impact in like bottleneck and the genetic diversity.
So you're suggesting that using the testers from the current pool, right?
Yeah.
The current individuals in the other pool is better.
This is the best because you are updating the frequencies, the current frequencies.
Yeah. I was gonna ask that question too, because even though that's the best you can do, and you said that between 3 and 5 testers are going to give you enough of a gain for all your accuracy and everything, but that's assuming that you can get, like the breeder wants to be using testers from the current pool.
Yeah.
So what
For
about
sure.
if you were to go back 5 or 10 years? Is 3 and 5 testers the right number? Is it actually more than that because you're at a different place?
Yeah, well, yeah, this is a situation. Breeders, because they like testers that perform well, like they know.
Well, one of the breeders
They don't want
are
to.
using testers from 20 years ago. So that's 20 years out of date.
Yeah.
Yeah, I think, well, I mean, the better question. I don't think the first problem for them to solve is, well, test blocking that.
No, I know. I know.
Yeah.
I think that they're getting anything, we're getting more testers.
Yeah.
It's going to be update your testers.
Exactly.
I've got a PhD student starting tomorrow. Actually.
Our breeders,
The first thing I'm
well,
going to read is
our breeders are implementing tests as part of testcrossing, but yeah, they are using tester that.
Well, that's the first.
Yeah. Well, didn't talk too much, but the tester also have some impact if this is an inverse line, if this is a single tester, single cross. But this is just the general trend, what we observe, and we can derive some from this.
I'm thinking why few testers is enough. And I wonder if the following analogy is a good analogy, because what we're trying to do is essentially try to estimate what the allele frequency is in the other pool.
Yeah.
And to estimate allele frequency in a certain population, you only need really a handful of samples, right?
Yeah.
that be a good thinking about why few testers is enough?
Well, this is what we observe. And of course, the diversity has to play a role there. We observe that few testers, we get, imagine if we have diversity and because we sample at random. And this is one thing. Of course, we need to avoid to choose related testers. Doing at random, it will job. You can do a better strategy for selected testers that on average will reflect the frequency.
If you're doing it, sorry to interrupt, if you're doing it at random, you do run with 3 testers, you do run the risk of actually choosing 3 very related testers too, just like you have the same risk as or reward of selecting very different testers.
Yes, and we're addressing this for the simulations, so you know, we run it 100 times and on average, it's gonna be fine. But we're saying that we probably shouldn't do it purely random in reality, but find representative individuals.
Yeah, that's.
Yeah, but it comes down to the question I said, if you're choosing from the pool, that's very low diversity, right? Then.
And just with that, we'll say thank you, David, for the presentation, because I